take a minute or so and then join us. So my name's Tamara Franz Odendahl. I'm the president of the Nova Scotia Institute of Science. So I welcome you all here tonight for our October monthly public talk. Today's talk is co-sponsored by WISE Atlantic, which is an organization that aims to break gender stereotypes in science to girls in junior high and high school and provides professional development opportunities to early career female scientists and engineers, and also seeks to promote the work done by female scientists in Atlantic Canada. So I'm delighted today to introduce you to Erin Bertrand, who earned her PhD in chemical oceanography from the Massachusetts Institute of Technology at Woods Hole in 2012. She went on to do a postdoc at the Scripps Institute of Oceanography and the J. Craig Venter Institute in San Diego, California. Dr. Bertrand joined Dalhousie University in the Department of Biology in 2015 as a Tier 2 Canada Research Chair in Marine Microbial Proteomics. Erin's work has taken her and her research group on over 20 ocean research expeditions, including 9 trips to the Antarctic Southern Ocean. Erin has received over $5.6 million in collaborative funding and $1.4 million in independent research fundings to support her research group at Dalhousie University. Erin was awarded the Dalhousie Faculty of Science 2020 Killam Prize, the 2018 Nova Scotia Discovery Centre Awards for an Emerging Professional, and in 2017, the Simons Foundation Early Career Investigator Award. Erin, welcome to the NSIS. We are really looking forward to hearing your talk. And your title is displayed, Phytoplankton Need Their Vitamins Too, How the Foundation of Ocean Food Webs Depends on Micronutrients. I'll hand the floor over to you. And we really look forward to your talk tonight.
Thank you so much, Tamara, for the introduction and for this invitation. I was really happy to accept and to share this work with all of you today. So my main goal for today is to explain to you how important these phytoplankton here are. So this is one example of a set of diatom cells. They're a type of phytoplankton. I'll explain to you why they're important, how they depend on vitamins for their growth, and how really the foundation of ocean food webs depends on this process. So I'll start by explaining that it's a microbial ocean. And I like to show this slide in my Department of Biology at Dalhousie in particular because most of my colleagues there study things like fish and whales that are up here at the top. This is a figure showing the percentage of biomass in the ocean and the different groups that make up that biomass. And as you'll see, the vast majority of the biomass in the ocean, we believe, is made up of single-celled organisms or microbes. So the vast majority of the biomass in the ocean is microbes with small contributions from things like fish and whales to this biomass. So this really motivates our study of phytoplankton in my group. In particular, we're interested in these algal autotrophs like these diatoms here, and bacterial autotrophs like cyanobacteria, shown here. These are particularly important because they fix carbon dioxide from the atmosphere and they can produce biomass that is then used for the rest of the ocean food chain. So we can see the scale of the importance of these ocean microbes here with this satellite image of chlorophyll A concentration. So this is a pigment that's produced by phytoplankton, and it's used, they use it to conduct photosynthesis. We can see it from space, and the high concentrations of phytoplankton are shown in green here, and lower concentrations in blue. So there's a lot we can learn from images like this. But what I really want you to take away from this for our purposes right now is that the scale of impact of these phytoplankton on the planet is massive. We can see them from space. The processes that they conduct are absolutely vital to many things that we care about. And I'll explain a few of those here. So this diagram shows us what happens when CO2 enters the ocean and photosynthesis is conducted by those phytoplankton. The material that phytoplankton produce in that process then can get cycled through all these different components we call the microbial food web. But importantly, much of this biomass ends up, or some of this biomass ends up going through the food chain to support the production of things that we care about for maybe our food intake, like fish. But importantly, some of this also can be processed by all these other microbes here and ends up sinking out of the surface ocean, and in the process, moving carbon from our atmosphere down into the deeper ocean. This turns out to be a quantitatively really important part of the ocean, or of the global carbon cycle, and has really helped the ocean absorb a lot of the CO2 that humans have put into the atmosphere. So these phytoplankton conduct really critical processes that matter for lots of aspects of our life, or of our lives. So what I'm interested in talking with you about today is what is controlling the growth of these phytoplankton. So we know that light is very important because they require that light for photosynthesis, of course. We know that temperature, up to a point, when temperature increases, it increases the growth of phytoplankton until some threshold. We also know that these phytoplankton require nutrients for growth, and that they can be consumed by predation, and they can be destroyed by viral lysis. Today, for the rest of our talk, the focus is going to be on these nutrients and how phytoplankton require them in order to grow. So when I think about the nutritional makeup of a phytoplankton cell, I think about what its cellular components are. So the nutritional requirements that phytoplankton have depend on the molecules that the cell is made up of. And I like this representation because it shows us just how important carbon, nitrogen, and phosphorus are inside a cell, and the different components that are important for making up those demands in a cell. So if we look at the protein pool, for instance, much of the nitrogen in a cell is contained in this pool. So if we want to know how much nitrogen a cell needs, we need to think about how much protein they require to grow. Similarly, if we want to understand how much phosphorus is in a cell, we need to be worried about how much RNA and how much DNA are there, because those are the major components of those are the major requirements for phosphorus inside the cell. So the sum total of all these biomolecules ends up making up the nutritional requirement for these phytoplankton. And in sum, on average, this requirement comes out to be about 106 molecules of carbon to every 16 atoms of nitrogen to every 1 atom of phosphorus. Now, I'm here to talk to you about micronutrients, and we have so far haven't mentioned those. And that is because micronutrients are required in extremely low concentrations, extremely low amounts that wouldn't show up on a figure like this, where we're looking at the percentage of cell mass that's made up of each of these elements that's required in much higher concentrations. So in order to understand micronutrient requirements, we need to look more closely at proteins, because these micronutrients are required mostly as cofactors for those proteins. And I'm giving you one example here of proteins that form photosystem 1. So this is one of the important cellular structures that helps the cell conduct photosynthesis. And in blue here, you can see all of these iron sulfur clusters. These are required for the activity of photosystem 1 and form, in fact, a large part of the requirement for iron in these diatoms. Another protein that requires a micronutrient I'm showing you here is called methionine synthase, or MET-H. And this enzyme has a demand for vitamin B12 or cobalamin, which is shown in these molecule structures here in black and blue and red. So in order to use this enzyme, diatoms or other phytoplankton, as well as humans, require the use of vitamin B12 to use this enzyme here. So the sum total of all of these micronutrients that are required for protein activity comes out to be something like this. So we've got our 106, 16 to 1.
Okay, you got it?
We've got our 106 to 16 to 1 for our major nutrients. And then we can see that very small quantities of metal micronutrients like iron and manganese are required. And an even smaller amount of that micronutrient, vitamin B12, is required for cellular growth. But it doesn't mean these micronutrients are not important just because they're required in small concentrations. And I like to think about this sort of as a recipe. So Phytoplankton are one of my favorite things. Chocolate cake is another one of my favorite things. But in order to make chocolate cake, we need to follow a recipe, and that recipe is going to have flour, it's going to have sugar, it's going to have baking soda and other things. So no matter how much flour or sugar you have, if you don't have any baking soda, you can't make your cake. The same goes with phytoplankton. No matter how much nitrogen and phosphorus you have, if you don't have any B12 or any iron, you just can't make your phytoplankton. So what we need to think about when we're thinking about whether or not the availability of these micronutrients is going to impact phytoplankton growth or impact our ability to make our cake, we need to think about the relative supply and demand. So if we have plenty of flour and plenty of sugar, but we don't have any baking soda in our cupboard, we're just not going to be able to make our cake. And the same goes for our B12 here. So I'll explain this in a little bit more oceanographic terms with this figure that I really like here. So this shows us the supply of nutrients, the supply of the elements that phytoplankton need to grow here on our y-axis. And on the x-axis, I'm showing you the demand. So this is the amount of each nutrient that's required relative to carbon in a phytoplankton cell, and the amount of that nutrient that's available in seawater as a ratio to carbon. And along this line here, we've highlighted this line because we know that nitrogen happens to be a limiting nutrient in much of the ocean. So if your element falls above this line, like these salts here, magnesium, sulfur, potassium, we expect these to never be limiting to phytoplankton growth. They're never going to run out of these elements in the ocean. On the other hand, if we look down here, where we can see the concentrations in cells of manganese, iron, and cobalt can sometimes be higher than the supply. So what this suggests is that this part of the graph here contains elements that are potentially limiting to phytoplankton growth in places in the ocean. So the average concentration or the average ratio of this element to carbon is shown in the dot, but the range is shown in this line here. So you can see for some of these elements, the range is quite huge. So what this means is that the demand for that particular element is flexible. So sometimes phytoplankton can grow with very, very little amounts of things like iron or cadmium or cobalt, and other times, and for certain species, that concentration that they require is higher. And I think one thing that's interesting to note here is as we go down here, as we go down this line, we get lower in concentration for this ratio to carbon, we start to see that this range is more dynamic. So the smaller the amount of a nutrient you need, the more dynamic that tends to be, and the more flexible that tends to be. That's a general pattern that we've observed. So this is one way to look at what nutrients are going to be limiting to phytoplankton growth. And another way to look at that is for us to go out into the ocean and do these quite crude experiments where we take water samples from the sunlit surface ocean. We separate that water that we gather into bottles that are transparent to the light that the phytoplankton need to grow. And we can amend those small samples of phytoplankton with different nutrients here. So for these bottles, we don't add anything. For these ones, we add nitrogen. For these, we add iron. And these ones, we add nitrogen and iron, just as an example. Then we put those bottles in these incubators that I'm showing you here, where we allow them to be subjected to natural light and the in situ temperature in which they were growing. Then we can wait a little while and observe what changes occur in that the amount of phytoplankton biomass that's produced. And what we can see here, when we've got our control where we don't add anything, we get a bit of phytoplankton growth. And then in the experiment where we add nitrogen, we don't see any growth. Where we add iron, we do see growth, and add nitrogen and iron, we see growth again. So what this suggests to us is that wherever this hypothetical experiment occurred, the ocean was phytoplankton growth in the ocean at that point was iron limited. So we can do these kinds of experiments throughout the global ocean, and we can start to accumulate lots and lots of data points to help us understand where in the ocean phytoplankton growth is controlled by what nutrient. So there's a lot going on in this figure. I'll step through a few key things I want you to take away from it. Shown in the background is phosphate concentrations.